Welcome to a microscopic look at the human immunodeficiency virus, or HIV. HIV is a retrovirus, a stealthy invader that targets the very core of our body's defense system.
Its primary target? The CD4 plus T lymphocyte, the crucial general of the immune army.
The virus travels through the bloodstream, searching for its host cell.
The moment of connection is precise and specific.
The viral envelope protein, GP120, first locks onto the character in image 1, CD4 receptor. This is where it attaches to a co-receptor, usually CCR5 or CXCR4.
This double lock signals the final stage, fusion.
The viral envelope merges with the cell membrane, creating a pore. The viral core, containing its genetic material, is released into the T cell's cytoplasm.
Inside the core, the virus carries its most essential tool, reverse transcriptase.
HIV's genetic material is RNA, not DNA.
The reverse transcriptase enzyme now performs a crucial step, transcribing viral RNA into proviral DNA.
This new character, in image 1, is now ready to hijack the host's machinery.
It leaves the cytoplasm and enters the cell's nucleus through a nuclear pore. Another enzyme, integrase, takes center stage.
Integrase cuts the host cell's own DNA and seamlessly inserts the viral DNA. The virus is now permanently integrated into the host cell's genome. It becomes a provirus.
The cell is transformed. It is now a viral factory.
The cell's own mechanisms begin reading the integrated viral DNA. These character in image 1 RNA transcripts serve two purposes: new viral genomes and messenger RNA for viral proteins. Ribosomes translate the viral messenger RNA into long polyproteins. These long chains are the building blocks of the next generation of viruses. The viral components travel to the cell membrane. Here, the assembly process begins. The polyproteins self-associate beneath the membrane patch, forming a curvature file.
The newly formed virus particle pushes outwards from the character in image 1.
This process, called budding, seals the new virion with a piece of the host cell membrane. The stalk separates, and the virus released is initially immature and non-infectious.
The last critical enzyme is protease. Protease acts like molecular scissors, cutting the long polyprotein chains into functional core proteins.
This cleavage allows the core to mature and transform into the characteristic cone-shaped capsid.
This cycle of infection, replication, and destruction severely depletes the body's character in image 1 count.
In the early stages, the virus multiplies rapidly.
The body tries to fight back, but the CD4 cells are overwhelmed.
In the clinical latency stage, the patient is often asymptomatic, but the war is still ongoing.
Millions of new virions are produced every single day,
relentlessly eroding the immune system.
When the character in image 1 and character in image 2 count drops below 200 cells per cubic millimeter, the diagnosis changes.
The patient has progressed to acquired immunodeficiency syndrome, or AIDS.
It's the onset of opportunistic infections.
These are illnesses that a healthy immune system would easily fend off.
Examples include pneumocystis pneumonia and Kaposi's sarcoma.
The key to managing character in image 1 is antiretroviral therapy TERAS.
ART is a combination of several drugs designed to block the virus at multiple points in its life cycle.
Nucleoside reverse transcriptase inhibitors, NRTI, are one cornerstone.
NRTIs are faulty building blocks that stop the proviral DNA synthesis.
Non-nucleoside reverse transcriptase inhibitors block the enzyme directly.
Integrase strand transfer inhibitors, INSTIs, prevent the viral DNA from merging with the host genome.
Protease inhibitors, Protease inhibitors PIs stop the final maturation of the virus. A PI molecule binding to the active site of the protease enzyme.
The virus released when PIs are used remains immature and harmless.
When taken consistently, ART can suppress the viral load to undetectable levels.
This concept is known as undetectable equals untransmittable, or UU. A person with an undetectable viral load cannot transmit HIV through sexual contact. While ART manages the infection, it does not cure it, as the provirus hides in resting CD4 cells. This viral reservoir is the main obstacle to finding a cure. In some cases, entry inhibitors can prevent the virus from docking onto the cell.
These drugs physically obstruct the virus's ability to lock on and fuse.
The goal of modern treatment is full immune recovery, allowing CD4 counts to rise back to healthy levels.
With continuous care, people with HIV can live long, healthy, and fulfilling lives. The battle against HIV also includes powerful tools for prevention.
One groundbreaking tool is pre-exposure prophylaxis, or PrEP.
PrEP involves taking 1 or 2 ART medications daily to shield healthy cells.
If HIV enters the body, the drugs immediately block the reverse transcriptase enzyme. This prevents the virus from establishing a permanent infection.
PEP must be started within 72 hours to prevent the virus from integrating into the host character in image 1.
Post-exposure prophylaxis, or PEP, is used after a potential exposure.
Both PrEP and PEP are highly effective when used correctly.
Safe sexual practices, including condom use, remain essential to prevent HIV and other STIs.
The use of sterile equipment is critical for injection drug users.
Mother-to-child transmission is now almost entirely preventable through ART during pregnancy.
Research is continuously striving for the sterilizing cure.
This involves eliminating the hidden viral reservoir.
Techniques like shock and kill aim to wake up the dormant cells and then destroy them.
Gene editing technologies are being explored to cut the proviral character in image 1 out of the host genome.
The development of a preventative HIV vaccine remains a major global health goal.
The complexity of the character in Image 1 envelope, which constantly mutates, presents a huge challenge.
Education is our most accessible weapon against stigma and transmission.
Understanding the science behind HIV empowers us to make informed choices.